the shape of the target site has changed, which prevents the antibiotic from being able to bind. So that's what we mean usually when we're over here, like alteration of target molecule. Yeah, that's usually what we're talking about. So a lot of the antibiotics where we have resistance, especially like protein synthesis inhibitors and things like that, you have a different shape, slightly different shape to the way that maybe the ribosome is put together, where it doesn't affect ribosome function, but it doesn't allow the antibiotic to bind. Yeah. Other questions? Yeah.
For synergistic effects and antagonistic effects, are those like for synergistic? Do they have the same? Because she was saying if you take them both at the same time, it increases the effect.
Right.
Do they both do the same thing and they increase each other, or are they doing two different things that complement?
Yes, I understand. You're asking basically, do they have the same mechanism of action?
Yes.
Or two different mechanisms of action?
Yes.
So usually when we're talking about a synergistic effect, so there's a couple things going on. So for example, here they've on this slide, you've got penicillin and streptomycin. So these are two antibiotics that have different mechanisms of action. So penicillin is a cell wall inhibitor, and streptomycin is a protein synthesis inhibitor. So just prescribing two antibiotics together is called combination therapy. And physicians and clinicians will often do this sometimes to help avoid antibiotic resistance, because, you know, if you end up having penicillin resistance, maybe you don't have streptomycin resistance, or, you know, you target them two ways, you can get rid of the ones that do have penicillin resistance because they don't have whatever. So lots of reasons you might do that. But the idea behind synergy that's a little bit, I guess, more advanced than just a combination therapy is that with synergy, they work together better than you would predict if they were like, you know, an additive effect. So normally, right, 1 + 1 is 2. So you're like, okay, cool, 1 + 1 is 2. But with synergy, 1 + 1 could be 4. Because they actually, like the expected activity of both of them is actually enhanced more than you think it should be. But, you know, with antagonism, you can see, again, two different mechanisms of action, but somehow when you put these two drugs together, they actually they work worse than you think they would. So 1 + 1 is like 1. Maybe there's no effect, but you're not getting the effect that you want to be getting. So, you know, when you test for antibiotic susceptibility, where's that? And you do, so I know like you do the disc diffusion in the lab, or you will do the disc diffusion, but when you do something like this, the broth dilution, which you don't actually do in the micro lab, depends on who teaches med micro, you may end up doing something like this in med micro, but there's a way to modify this assay where you can use two antibiotics and you can determine if there is synergy antagonism or just a combination effect. So yeah, and you know, one of the other combinations I think that they highlighted is, how many of you have ever taken Augmentin, or you've been prescribed Augmentin? No? How about amoxicillin? There's a lot of resistance to amoxicillin, so now usually they won't even prescribe you just straight amoxicillin, they prescribe you Augmentin, which is amoxicillin plus a drug called clavonic acid. That's a beta-lactamase inhibitor. So it inhibits the enzyme that would destroy amoxicillin. So technically the bacteria are still resistant to amoxicillin. They produce an enzyme that can destroy amoxicillin, but you take it in combination with a drug that inhibits that enzyme, so now you can overcome the resistance. So a combination therapy doesn't necessarily have to be two antibiotics. It could be the antibiotic and something that helps you to overcome or prevent resistance. And Augmentin is really the classic example of a combination therapy that isn't two antibiotics, it's an antibiotic plus another compound. Other questions from chapter 20? Yeah.
For like the different kinds of like more general information that was on the slides, like, oh, in hospitals, like these contributions to antibiotic resistance, doctors have to do these things, these are like the responsibilities. Is that something that we'll need to like keep in mind General consideration.
I want to make sure that I pull up the slide you're talking about so that I don't answer the question incorrectly. Like this one?
Something like this one, yeah.
I think it's really important for just you to know as an informed graduate in the biological sciences, but especially for any of you considering going into healthcare fields. This information is absolutely critical. Because, and I don't know if this statistic is in here. Oh, yeah, here, that if we can't address this issue of antibiotic resistance by 2050, it's estimated that antibiotic resistance will kill 10 million people a year. That's how many people cancer kills every year. So it'll be just as bad as cancer. And we have already had infections in the US where patients have an infection that is resistant to every antibiotic you could possibly get in the US. And when I teach a class like Med Micro, I always pose this question to them. So how do you treat your patient? Your patient has an infection with a bacterium that is resistant to every antibiotic. And they always tell me, well, combination therapy. I'm like, well, it's already resistant. It's still resistant. It's resistant. There are 26 or 27 FDA-approved antibiotics. It's resistant to every one of them. How do you treat your patient? What happened to that patient? You don't treat your patient. The patient dies. So when a patient has an infection that cannot be treated with antibiotics, we don't have anything else. That's why it's so critical for any of you planning to go into healthcare to be really cognizant of course, you don't want to give your patient an infection, right? That's bad. But you don't want to give them an infection with a antibiotic-resistant bacterium, which is really common in the clinical setting because we don't have anything else. That's what they do. Like here's the other option. So when patients get infections, things like the necrotizing fasciitis, that can be caused by lots of different bacteria, but it's the flesh-eating infections. You start removing the limbs. It's in the feet, you take off the leg. But it's probably already spread to the other leg, so then you have to take off the other leg, and the fingers, and the hands, and the arms, and then the patient still dies. So you either remove the infection by removing the body part, or your patient dies. So it's really critical that you keep your patient healthy as best you can, that you as a healthcare professional are a good steward of antibiotics, even if you're not a healthcare professional. I'm not a healthcare professional, I'm just a professor, right? Yes, you have to call me doctor, but I'm not that kind of doctor. Sometimes, if I look at my bank account, I'm like, I wish I was. That would be cool. That'd be cool. But also, you people, blood, guts, blah. But I have to be a good steward of antibiotics. This is my job. I don't want you to leave here and kill your patients. I want you to understand why these things are so important. And that's me going to the vet and you're going to give your dog these antibiotics and you have to give them the whole I'm like, girl, I know. And then my vet finally remembered that I teach Med Micro, so she was like, okay, no, I got you. But the vet has to do it, right? Because what was the statistic? 80% of all antibiotics are used in animals. Okay.
So with technology becomes so good nowadays, CRISPR and all that, is that gonna help the whole antibiotic resistance by changing genes and all that? Or?
It would probably take too long because these infections, like a septic infection, you're talking hours to days.
But for overall,
oh, they'll just continue to evolve. So you might treat this patient and their infection, but it's still living in the hospital. So the best alternative therapy that's starting to be researched right now are using bacteriophages. Because bacteriophage don't hurt the host, they kill only the bacteria that they infect. But the challenge is you have to know exactly what infection that is because you have to find the right phage that will kill that particular infection. You have to get it to where the infection is and you have to make sure it gets cleared out. But as of right now, that's kind of the last hope. Yeah. Other questions regarding this material? Anything? Yeah.
I guess this is more general for like what's going to be on the test, but would we have to know like the mechanisms of the antibiotic resistance, like being able to draw it out and kind of listing it like we did the lab station?
I would think that it would be very important for you to not only know the major mechanisms of how antibiotics work, but yes, the major mechanisms for how cells become resistant to antibiotics. Yeah, I would find that very important. Yeah. And you know, one of the final things to kind of emphasize here is that you know, humans did not cause antibiotic resistance. It wasn't like all of a sudden there's penicillin and now there's penicillin resistance. There's always been penicillin resistance. There's always been antibiotic resistance because antibiotics weren't something that we created either. Microbes have been living together, especially like in soil communities, for millions or billions of years. They don't really care about us. They have been competing with each other for space and resources for billions of years. So one microbe evolves a mechanism to kill the neighbors so that it can have all the resources. Well, the neighbors eventually will evolve a mechanism to not be killed. That's resistance. So we don't cause antibiotic resistance. We just make it very bad. We make it so much worse because we overprescribe. We don't take antibiotics correctly. We don't follow treatment regimens. You call the doctor, oh, my kid has a runny nose. Well, here, take some amoxicillin. Dude, your kid has the flu. Every kid has the flu. Right now, all the kids have the flu. Antibiotics don't do anything for the flu. The flu's a virus. Antibiotics don't do anything for COVID. COVID's a virus. They don't do anything for measles or chickenpox or any of those things. I feel better. I'm not going to take my antibiotics anymore. Cool, you just trained all the bacteria still left in your body what to do. That was cool. Good job. I'm going to go to another country and just buy them, so just in case, right? Just because you can do things doesn't mean you should. So it's really important that we are good stewards of antibiotics because it just makes it worse. And that's my favorite video of all time. So, okay. Any other questions? Oh yeah, my comics. Okay, cool. Well, if anything comes up, feel free to email me, let me know. Or come to my office hours on Monday. So let's go ahead and jump into microbial genetics. How many of you have or are taking genetics? Some. Okay, cool. So a lot of this will be a review of concepts you learned when you took genetics or of concepts you are learning as you're taking genetics. Those of you who haven't taken genetics yet, this will serve as a foundation. So when you do take genetics, you'll be like, yay, I remember these things. Okay. So when we talk about microbial genetics, we can talk about the prokaryotic chromosome. We're mostly going to focus on prokaryotes because this is microbiology. But the nice thing is that if you understand the foundation of what's going on in the prokaryotes, you can often apply a lot of that to what happens in the eukaryotes. We do things very similarly. We just like to add extra complexity. So again, just as a review, most prokaryotes have a single chromosome. It is almost always circular. It does not exist in a nucleus. It is supercoiled, like really tightly wound up and packaged within the cell. So what you're looking at in this image is a cell of E. coli that has been exploded and the chromosome has been pulled out. The length of the E. coli chromosome, they said, is about 1,000 times the length of the cell. Same thing with our DNA, right? If you actually extended out our DNA, like end to end, it's really, really long. E. coli was one of the first, but not the first, bacteria that was completely sequenced. We have the whole genome sequence of E. coli. So you could represent it in a circular map like that. It's approximately between 4 and 5 million base pairs. It depends on the strain. Like the lab strain that you work with all the time, it doesn't have antibiotic resistance, it doesn't have any real virulence factors. But then there's something like, you know, E. coli O157H7 that has a lot of virulence factors, may have antibiotic resistance, the genome might be larger. So really the first thing that we talk about when we talk about genetics in any system is how the genetic information flows. And so, you know, we'll talk about all three of these concepts, but when we start with a bacterial cell, right? I mean, we go all the way to the right, we could talk about replication. A bacterial cell, just like any cell, has to replicate its DNA. And then when it divides, each new cell gets a copy of that DNA, replication. Very commonly in cells, we also do expression of genes. We'll talk a lot about, or you'll hear people talk about gene expression. So what that means is that that genetic information held within the DNA is being transcribed into RNA and translated into proteins. Gene expression. Yeah, eukaryotes do the same thing. We all do transcription and translation. Maybe we do them a little bit differently. Maybe eukaryotes make it a little bit more complicated. They do, we make it more complicated. But the same basic processes are the same. Now, where prokaryotes kind of stand out and do something different compared to eukaryotes is this middle term, recombination. Bacteria are able to share genetic material within the same generation. Eukaryotes don't do that. If you want to share your genetic material, you need to have a child. That's how you share your genetic material. Your genetic material does not go to another individual until you reproduce another individual. But bacteria can be like, hey neighbor, here's a piece of DNA. You want it? And you know, if we use a silly example, my daughter has blue eyes, which I think is really weird. I have brown eyes, but I'm like, girl, I like your blue eyes. Can I have your blue eye allele too? I want blue eyes just for funsies. But she can't give that to me. She would if I asked her. Her sister has brown eyes, so she gets very upset that she has blue eyes and her sister has brown. So I mean, I can't trade that with her. She got what she got. But if I was a bacterium and I'm like, hey, you're antibiotic resistant, but I'm not, can you help me out? I mean, they don't ask, they just do it. And so they can share properties, especially things like antibiotic resistance, with members of their own generation. So that's a really cool thing that bacteria can do. Okay, so again, I've already talked about this. You go from DNA to RNA to protein, and you might have heard this term before, the central dogma of molecular biology. DNA is transcribed into RNA, which is translated into proteins. And I think it's funny that we call it dogma. To me, like dogma has a kind of religious connotation, like it's the rule, it's right, that's the dogma, it's the rule, there's no exceptions. Except there are exceptions. There are organisms that can turn RNA back into DNA, so maybe we shouldn't call it the central dogma anymore. But that's what we call it, that's what it means, that's the central dogma. I told you about this idea that microbes can share bacteria with members of the same generation. We call that horizontal gene transfer, and we'll talk more about that at the end of this chapter, the mechanisms by which that occurs. And then, of course, the boring one, vertical gene transfer from one generation to the next. And in bacteria, that's as simple as I've copied my chromosome and made 2 cells. So they basically clone themselves, which is different from how multicellular eukaryotes tend to reproduce. Most of them. Okay, so what happens during replication? This is the first area that we're going to go into some detail on. And again, I know this is not everybody's, unless you're into genetics, it's not everybody's favorite part of microbiology, but bear with me. It's really foundational, super important, And we will get into things that will make you bleed out of your eyeballs later. Okay? Promise. I promise. I will not let you end this semester without like tapeworms and weird stuff. Okay. So DNA replication is this idea that we have, as a bacterial cell, a usually singular chromosome that we want to make a copy of so that we can make a new, two new cells, right? The two daughter cells. We have a lot of enzymes that are involved in this process. There are a lot of details that are really important for how this process works. But again, for those of you who've already taken genetics, you've talked about this in a lot of detail. I know it's something that's usually covered in some of the intro biology classes too. So hopefully a lot of it's review, but maybe it'll, brush up on some of those concepts that you were like, well, maybe they'll become a little bit more clear. So we have double-stranded DNA as cellular organisms. Things like viruses can have single-stranded DNA, but we're cells, we have double. We can use both strands of the DNA as a template. Okay. So that means that each strand of DNA can serve to be copied for making the new strands of DNA. We use an enzyme, and don't worry, we're going to have a lot of slides that have these enzymes on there. We use an enzyme called DNA polymerase to add the new nucleotides to make our new strands of DNA. But there are things that we have to think about before we can even start replicating. One of the challenges with DNA in the prokaryotic cell is that it is really tightly wound in those supercoils. But it's also double-stranded. The two strands are bound to one another. How do we get an enzyme in there so that it can see the template and make the copy? So in order to do that, we use an enzyme called helicase. Helicase is capable of basically breaking those hydrogen bonds to separate the two strands of DNA. So we got to separate the strands of DNA so that we can get all the machinery in there. But one of the things that happens just because of that coiling of the DNA is as you start to unzip it, it's like if you're trying to separate, if you ever have like a necklace, like a really thin chain that gets all tangled and you're like trying to pull it apart and it just makes the knot worse. Right? Or if you knit or embroider whatever, thread, necklaces, they all get tangled. Then you try to open them up and it's a hot mess. Well, DNA does the same thing. It's a hot mess. And at some point you're stuck because you have this big knot that you can't do anything with. So we use an enzyme called DNA gyrase to relax the supercoils. So that allows helicase to continue opening up and spread that replication fork so that we can continue to replicate. So these are kind of the first things that have to happen. You have to be able to open up the DNA, and both of those are involved in helping to open up the DNA. And remember, if you don't have DNA gyrase, you don't get very far. You have that big mess, knotted mess of DNA. So this is an antibiotic target. Ciprofloxacin, if you've heard of Cipro, and the derivatives, inhibit DNA gyrase. It's a great target for antibiotics. So these are what some of the antibiotics that inhibit DNA replication, that's where they do it. You can't open your DNA, you can't make a copy of it, you're done. Okay. So some of the other things that are pointed out on this particular slide Nucleotide bases are added in the 5' to 3' direction. I don't know why, but when I was an undergrad, this infuriated me because I didn't understand it. I was like, I could never, like, when I'm looking at the DNA, I'm not a spatial person. Like every whatever standardized test, that's the part I would fail. If you rotated this and tried to fit it, and I'm like, I don't know, I don't care. Okay, so this didn't make sense to me. But now it does. So the idea here is that when you're looking at a molecule of DNA, right, you've got your sugar, here's your base, like whatever the part of it is, right? This means it's ribose, or RNA, this is DNA, either or, right? Either or. And we number the carbons, right? 1', 2', 3', 4', and then there's like one that sticks out here, 5', and then you've got phosphate group. So this is base number 1 of my new chain. So when we say that DNA is added in a 5' to 3' direction, what we mean is that when base number 2 comes in, this 5' end of the new base joins with the 3' of the previous. 5' of the new to the 3' of the previous. So that's what we mean when we say 5' to 3' direction. We don't know of any system that could add a base up here at the front. You can't do that. You can't cut the line. And I always say, like, we don't know yet. Maybe one day we'll find a very weird life form that has very weird enzymes that could potentially do that. But as far as we know, there is nothing that cuts the line. You have to add to the back. And you have to have this free hydroxyl group, the OH group. Now, as I know most of you are biology majors, so as you continue through your biology career, you have options to take. You have to take a genetics and biotech class. Maybe you'll take genetic biotechnology, where you talk about DNA sequencing, which takes advantage of this 3' OH. Okay. So it's really important to understand that 5' to 3' means the 5' of the new base binds to the 3' of the old. And then base number 3 would come in, and the 5' end would bind to the 3' end of the old. 5' to 3', 5' to 3'. One of my students gave me a sticker, and it's DNA replication, and it says working 5 to 3, what a way to make a living. And I was like, so I haven't put it on my door yet, but that's, the kind of like science dad jokes I like. But that's what it is. You're working 5 to 3, 5 to 3, 5 to 3, 5 to 3. No, it's bad. I know a lot of science dad jokes, so I apologize in advance. Because they'll come out whenever I have an opportunity. Okay, so that's what we mean by 5' to 3'. And if you're still like, yeah, that still didn't make any sense, totally cool. Please come talk to me. I want to make sure that everyone who wants to know this knows this. Okay, so if you want to learn it, come talk to me and I will help you learn it. Okay. The other thing that's mentioned is this idea of needing an RNA primer. So I told you, so you have an enzyme called primase. That's the one that makes the RNA primer. So why is that important? Why is the RNA primer important? Okay, you have to add 5' of new to 3' of old. 5' of new to 3' of old. We use an enzyme called DNA polymerase. Oops, I can't spell today. Or most days, it's fine. right? 5' to 3'. DNA polymerase is great, and we'll talk about all of the different cool things it can do, but it can't do this one thing. It doesn't know how to start. It can't put base 1. It's like, oh, yeah, I don't know. If there is a base, it can add a new one. It does not know how to start from scratch. You gotta give it something. So this seems very inefficient. DNA polymerase can't even start replication because it can't put down base number 1. Instead, we use an enzyme called primase, which is a type of RNA polymerase. And it puts down some RNA bases, maybe 10 or so. And then DNA polymerase is like, oh, now I know what to do. It doesn't know where to go. It doesn't know how to start, but RNA polymerase does. It has figured this out. Well, actually, it knew already because that's more of an ancestral thing. DNA polymerase forgot how to do it. So RNA polymerase has to say, this is what we do. Here's where you go. Here's your free 3'. And DNA polymerase can come in and start adding the bases. So on this slide, I mean, they only mentioned 3 of these enzymes, but I added the 4th. There you go. So, and at any point, if you want me to stop and explain something, please feel free to raise your hand. And if I haven't seen you, because actually I can only see close things with my glasses, you can like yell at me too. That's fine. I don't know if you knew that. I can't really see you in the back. Sorry. I know you're there, but sometimes I can't see the hands. Okay. This is, whoa, right? It's everything all put together. And we'll come back to this. But let's start basically at the beginning. So at the beginning, helicase separates the DNA strands. And I don't love this figure from the textbook because it doesn't really put in the DNA gyrase right away, but that's fine, whatever. So you unwind the strand and separate the DNA. DNA gyrase helps to relax the supercoils. Primase puts down the RNA primer. Perfect, that's great. Okay. Where does this occur on the chromosome? This occurs, at least in bacterial chromosomes, at a region very creatively known as the origin of replication, because it's where replication starts. Origin of replication. So it's a particular sequence of DNA that signals for these things to come in and bind. For linear chromosomes, like ours, we tend to have multiple origins of replication, but a bacterial chromosome will generally have 1. Okay, so what happens next is, and again, these don't necessarily happen like 1 and then we wait for 2. Sometimes these things are happening kind of all at the same time. So DNA polymerase, once it's open, primase has added the primer. DNA polymerase can start adding the bases, but now you have this single-stranded DNA that wants to come back together because it's like, well, no, there's my friend. Right? They want to come back together. So you use what are called single-stranded binding proteins, which I'm never going to spell out again, because I will call them SSBPs. They keep the DNA open. So they keep the DNA from closing back in on itself. Okay. And they've illustrated those here. Okay. So now this was the other part of DNA replication that used to make me irrationally angry. I don't know why 5' to 3' made me so mad, but it did. And Dr. Youngbloom was very kind when I would sit there and glare at him. Because eventually I got it, right? He knew I would, it just took me a minute. Leading and lagging strands, for some reason, just made me very upset. And because I felt like, oh, just go, like go. Well, they can't, right? Because they are limited by the fact that they have to work in a 5' to 3' direction. And what we have done, including bacteria, so I'm blaming them too, we have set up our DNA anti-parallel. Which means if you have one strand, 5' to 3', its paired strand, 3' to 5'. They are anti-parallel. So maybe I'm just mad at evolution. I'm not mad at the fact that this is what happens. I'm mad that we have to talk about it because you could have made it simpler for me. But why? Why would they do that? Okay, so that means if I can only add in the 5' to 3' direction, I'm going to copy these two strands, right? Oh, I'm not supposed to use this kind of marker. They'll yell at me. We get nasty emails every few weeks. Don't use expo markers. Somebody brought a bunch of them in here. Right, so my black strands are my new strands. This one has to be 3' to 5'. This one has to be 3', 5'. Okay. So I have to have that free 3' end. So I'm going to get my primer is going to be here, right? It's going to be 5'. Finally, I'll get my 3' in, and I synthesize this strand in this direction. Okay. Here I've got my 5', and then this is my little primer. Here's my 3' into my primer, and then I synthesize in this direction. So the challenge is I want to copy both of these strands of DNA, but I have to do them in opposite directions. If I'm opening up the DNA this way, I can copy one strand in this direction, and I can copy the other strand in this direction. So, why is that a problem? Who cares? Okay, well, this one's not a problem. We opened up. Here's my 5'. Here's my 3'. So I would put my, primer right here, and I just start moving. Right? 5', 3', 5', 3', 5', 3'. And as the DNA continues to open, I just keep moving towards the replication fork. I don't have to stop, right? It's opening. Oh, free 3', 3'. We call this the leading strand. It is synthesized continuously in one piece. It will continue to move toward the replication fork as the DNA is opening. The one that's ugly is the one in the other direction. Now, in the other direction, right, I have 5' to 3'. This is supposed to be my new strand. Right, my new strand, 5' to 3'. Okay, and so, all right, this part's open. I can add my primer and start synthesizing. But then now this part opens. So, okay, I have to add my other primer. And then this part, oh my God, right? Over and over and over. As the fork opens, I have another little spot I could add a primer. And then I go back to my first primer. And then I go back. And then I go back. And so what we say is that that one is synthesized discontinuously. Because it's not synthesized in all one piece. It's synthesized in these little chunks. And these little chunks that are synthesized that way are called Okazaki fragments. They're named after a scientist whose name was Okazaki. He discovered them. And this is the lagging strand. Okay, so we still haven't come into any new enzymes yet. So we have leading strands, we have lagging strands. We have RNA primers everywhere. So what do we do with all these RNA primers, right? We know that DNA is DNA. There aren't chunks of RNA within our DNA. Okay. Well, DNA polymerase can also remove the RNA primer and fill with DNA. It's a multifunctional enzyme. And you can do that on both the leading and lagging strand. The leading strand will only have like one primer. That's cool. The lagging strand has all the primers. Great. Now there's still a problem because what happens is when the primers are digested and the new bases are added, there's a gap in the backbone itself. The DNA polymerase doesn't seal the gap in the backbone when it replaces the primer. So instead, we have an enzyme called DNA ligase. It seals the gaps in the backbone. And then you're done. So you have a number of enzymes, you have a lot of proteins, and then you have, those two leading and lagging strands. Okay, questions to this point about DNA replication? Theoretically, I could find it very important that you be able to draw a replication fork and describe the enzymes and proteins involved in the process. In theory. Question?
My question was going to be, how much of this do you have to know for this class?
Yeah, okay. Yes, I could find it very important that you be able to draw a replication fork and label and explain the proteins and enzymes involved in the process. Again, this is one of those, like, not even micro things, it's like foundational biology things. Yeah. You'll see it in lots of classes. Or if you've already taken genetics, maybe this was the last time you see it, but you see it. So, some of the other things to think about with replication, we know that when we're talking about joining the 5' and 3' end, we're joining the 5' phosphate group to the 3' OH group on the sugar, which is either deoxyribose or ribose. And that costs energy to make that bond, so it is energy intensive to make a new strand of DNA or to make a strand of RNA. And usually what happens is, as the nucleotide is being brought in, so here it's bringing in a thymine, it's a thymine triphosphate. Because remember, ATP and things like that are still nucleotides. So it has the 3 phosphates, so it gets the energy from breaking those phosphate bonds, and then has the energy to form the phosphodiester bond. Okay, the other term on here is that DNA replication is semi-conservative. We're not talking politics, so what does that mean? What is semi-conservative replication?
Like it preserves half of the original?
Yeah. So, if this is my original strand of DNA that I have replicated, here's my new strand of DNA. This one goes to this cell. This one goes to this cell. Each cell has a molecule of DNA that is half old, half new. We have conserved half of the DNA, semi-conservative. When you take genetics, you'll have to know the experiment that allows you to determine that. But not for this class. So that's what it means by semi-conservative replication. actually a very elegant experiment. If you've taken genetics and you don't remember, it's the Penicillin and Stahl. They used heavy radiolabeled nitrogen to determine that. Okay, so specifically what happens with bacterial DNA? This. So again, step by step, bacterial DNA replication starts at the origin of replication where this process happens, right? You use helicase, you use DNA gyrase, you get the primase. But the cool thing about bacterial replication is that it is what we call bidirectional. So at the origin of replication, and here they've moved it up here in this picture, the DNA opens, right? And it opens in both directions, and the replication proceeds in both directions. So it's not like it starts at the top and then goes clockwise or counterclockwise or whatever. It starts at the top and moves in both directions. So it takes half the time, right? If you had to go all the way around, it would take much longer than if you only have to go half the way around. So it goes half the way around, and it ends at a site called the termination site. So all the machinery will fall off, and you'll now have 2 separate chromosomes. Bidirectional replication. Okay, questions about DNA or replication? Okay, sweet deal. All right, transcription. Okay, what are the major differences? I mean, they're listed up here. What are the major differences between DNA and RNA? You guys can read, but I will summarize them for you as well. RNA has ribose as the sugar instead of deoxyribose. So remember, that means it has the OH group at the 2' carbon, where DNA has just the hydrogen. The 4 bases that make up DNA are A, T, G, C. The 4 bases that make up RNA are A, U, G, C. RNA uses a uracil instead of a thymine. The other major difference that we tend to see in our cells, again, our cells, is that RNA is usually single-stranded. Usually. Double-stranded RNA in our cells often signals a problem, and our cells have a lot of mechanisms for destroying double-stranded RNA. And the reason that's a problem is, well, one, there are a lot of viruses that have double-stranded RNA. So if our cell has double-stranded RNA in it, that could mean it has a virus. So we got to get rid of it. But it's also how we can target RNAs for destruction. And then, of course, very commonly, our DNA is found as a double-stranded molecule. So the process by which we use DNA to generate any kind of RNA is called transcription. So we're taking the DNA message and turning it into an RNA message. I guess we always think about this like in the context of gene expression, right? DNA to RNA to protein. But there are a lot of RNAs we might make that don't necessarily like encode for the protein. But again, classically, that's how we think about it. So as with DNA replication, RNA polymerase also only adds bases in the 5' to 3' direction. That's the limitation of polymerases. Again, we've never found a polymerase that can add in the opposite direction. It has to be 5' to 3'. Usually for transcription to start, there's a region of DNA that's called a promoter. The promoter is going to have a specific sequence that will bind to the RNA polymerase. So the RNA polymerase can recognize the promoter region on DNA and know that that's where it's supposed to bind in order to start doing transcription. There will be a sequence at the end that is the terminator that says RNA polymerase can stop and no longer needs to add more bases. And, you know, as we know, mRNA is the major form of RNA that we talk about because it's the template for doing protein synthesis. And so, you know, sometimes, I don't know if you've ever wondered, but like I would wonder, why do we even do this? Like, why do we go from DNA to RNA? Like, why can't we just go from DNA to protein? Like, why do we have to have this weird intermediate? And it's not that different from RNA or from DNA. Like, what's going on here? Well, evolution doesn't have a why. Evolution just is, first of all. It's just kind of annoying. But I like to try to explain things to myself anyway to make it less irritating. And, you know, one of the things to think about is there's a lot of stuff in DNA that we don't necessarily need when we're doing things like making a protein. Do I need the promoter sequence to be turned into RNA? No. If I'm talking about a eukaryote, do I need intergenic regions to be turned into RNA or proteins? Well, no. Do I need introns to be turned into proteins? No. And so an analogy that finally clicked for me maybe in the past 5-6 years, you ever tried to get a recipe on the internet and you got to read like somebody's life story and then like where they bought the like organic free range flax or whatever that they were, I'm like, oh my God. Right? And then they finally give you the button that's jump to recipe. And that's the part that you copy and print. That's basically what you're doing. You're trying to, as much as possible, focus on the part of the DNA that you need. And it's going to look a little bit different, but not that different from the original instruction. I don't need the preamble of why you and your children are very excited about eating cake. Cake is awesome. Like, of course you like cake. I don't care, right? But I want to know how to make the cake. So that's kind of what we're doing. Or maybe, you know, you've got grandma's recipe book, and if your grandma was anything like mine, she learned how to write shorthand. I'm like, Grandma, please. Please, Grandma. So when I, you know, I'm like, okay. But even her recipes, right, they're a little bit harder to read. I can write them out so that I can understand it. So you're taking a message and just turning it into a slightly different message that will help you get the product you want. That's transcription. So we have the promoter where we start, and this is upstream, so it's before where we actually want to do transcription. So these enzymes are actually like complex enzymes. It's not just like a single enzyme. It's like a complex of proteins, and they're kind of big. So they bind what we call upstream, which means on the DNA just before where we want to be. So if this is the first base that I want to transcribe, my promoter has to be over here in front of it so that my promoter has enough room to sit all the way, or so that my polymerase has enough room to sit all the way down and start doing transcription. RNA polymerase has helicase activity. It can unwind the DNA. Usually we're not terribly worried about things like supercoiling because we're talking about maybe at most 2,000 to 3,000 bases that would be transcribed at a time. We're not talking, you know, millions of base pairs here. Most, especially prokaryotic genes, are very small. But then much like we see with DNA, you have a little replication fork. But instead of trying to synthesize from both strands, we're using only one strand as a template. So we have just the leading strand, right, being synthesized in that 5' to 3' direction. And we're just adding the RNA bases, 5' to 3', 5' to 3', 5' to 3'. Okay. There aren't like single-stranded binding proteins holding it open. Once the transcript, right, the piece of RNA has started to be synthesized and the polymerase moves out of the way, the DNA will just come right back together. And then it reaches a terminator, which is usually another sequence, and everything falls apart. And you now have a free molecule of RNA. It's a lot simpler than replication. I'll give you the hard one first, and then we do like some of the easier ones. That's transcription. And if we talk about transcription in the context of, well, now we want to make a protein, we talk about translation. Now this is where you go from, you know, the like weird printed recipe on the internet to the one that maybe I wrote down or put on my phone or whatever so that I could like easily see the recipe. So you go from one version of a recipe to a different version of a recipe to the actual food, something very, very different. So you're going from DNA or RNA, which are sort of the same, similar enough, to amino acids, which are not at all the same. So when we do that, we have to read the RNA in a particular way. It's very fussy. You have to read it in groups of 3 letters. We call them codons. So we read if you're a band person, you'd be like 1, 2, 3, 4, 5, 6, right? One of those people. I was in band too, I get it. Right, you read them in groups of 3. And each group of 3 letters of RNA represents 1 amino acid. One of the things that's very consistent among systems is that AUG means start. So if the RNA is AUG, the ribosome goes, oh, this is the beginning. There are various stop codons. We call them nonsense codons because they don't give a message. There's no amino acid that comes in. It's just done. Everything falls apart. Literally, everything just falls apart. So again, that language of converting RNA to amino acids is the genetic code. And I would never expect you to memorize a table like this. I don't know this. Half the time I can't remember all 3 of the stop codons. Like, you gotta remember the start codon, like please remember the start codon. AUG, start codon. Pick a stop codon to remember. I always remember UGA. I don't know why, but that's the one. But you do have to be able to know how to use a table like this. Does everybody know how to use a table like this? Yes, no, maybe so, yeah. I mean, you've seen it before. So we have 64 possible codons, right? Because each codon has 3 letters, or 3 positions, right? First, second, and third. Each position can be filled by one of 4 letters. So you have 4 letters that could go in spot 1. Be careful here. 4 letters that could go in spot 2. 4 letters that can go in spot 3. 4 times 4 times 4 is 64. So we have 64 combinations. 61 of those combinations are sense codons. They'll give us an amino acid. And then 3 are nonsense. They give us nothing. We call them our stop codons. And then we have 20 amino acids. And so, if you needed a refresher about the table, and you're thinking about a codon, I'm just going to randomly pick one. Okay. If you didn't remember how to use the table, or you didn't want to say anything like, I don't know how to use the table, that's fine. You start here on the left, first position. So the first position in my codon is A, so I'm somewhere in this whole row, right? I'm in this row. All of these codons start with A. My second position is G. So I'm in this column. So this column and this row intersect. Now I have 4 options. My third letter is U, which is right here. So AGU is the amino acid serine. So that's how you read the table. So theoretically, and I'm not promising anything with this one, this really is a theoretical. Theoretically, if I gave you codons, right, I gave you a chain of RNA bases and I gave you this table, you should be able to make a protein for me. Again, I would never just give you the RNA and be like, good luck, make the protein. I've got to give you the table. So I would give you the table and you should be able to make the protein. One of those fundamental biology things. But what's cool about the table, I guess, cool about the biology behind the table. The genetic code is what we call degenerate. And that's because, remember, we have 61 possible codons, not counting the 3 stops. We have 61 possible sense codons, but we only have 20 common amino acids. There are 22, but that's fine. 20 common amino acids. What that means is that for a lot of amino acids, there are multiple codons that could give you the same amino acid. So why is that important? Well, that's important because it allows you to make a mistake. By you, I mean DNA. It allows there to be a mutation in the DNA that theoretically could give you the exact same amino acid. Boop. Okay. So if I had a mutation that in my template strand, right, my template strand, GGG, okay, CCC, whatever, proline. But what if I had a mutation that instead, if I were looking at the strand, it's GGT? CCA. Still proline. And I can make a mistake and it's okay. Sometimes you can't make a mistake. Methionine does not let you make a mistake. Okay. But oftentimes you can. And sometimes you can even make a mistake and get a different amino acid, but it's really similar biochemically, so that's nice too. But I think it's methionine and, if I'm not mistaken, methionine and tryptophan are the 2 that are very unforgiving. You get 1 shot. So the degeneracy of the amino acid code is really awesome because it allows for mutations. Not at every site. Most commonly at the 3rd site of the codon, you could have a mutation and still get the same amino acid, which is great. Our system has evolved to tolerate mutations because mutations occur all of the time. So, of course, the mRNA is very important for translation because it is the recipe that we're going to read in order to bake the cake. tRNA is another really important RNA molecule involved in translation. It is transfer RNA, and it brings the amino acid to the ribosome so that we can start making the protein. So it has something called the anticodon. So tRNA is like this kind of clover-shaped molecule that'll have an amino acid at one end. I made it methionine. And then if my mRNA, right, for methionine has to be AUG, my anticodon UAC, because they have to be complementary. Right. They're specific. So you have a tRNA that has, this is called the anticodon, because it's on the one that's on the tRNA is the anticodon. And it, in theory, should be complementary to the codon. Okay, so I want to stop here, and what I want you to do now with the last 5 minutes is in your groups, and because we haven't talked about it in a while now, what are the 5 major mechanisms or sites of antibiotic action, and what are the 5 major mechanisms of antibiotic resistance? So in your groups, put your name down, take about 5 minutes, and then you're free to go. If it takes you less than 5 minutes, you're free to go.